Welcome back to the Deep Dive. Today we are going to take a stack of some seriously dense academic material, crack it wide open, and give you the vital knowledge you need.
We certainly are.
And today we are diving deep into the microscopic world of microbial secondary metabolites. These are the true chemical powerhouses that really underpin the entire biotech industry.
That's a great way to frame it. Our mission today is to move beyond, the simple penicillin story everyone learns.
Right.
We want to explore the entire industrial landscape, how we discover these compounds, the really intricate mechanisms they use to attack pathogens or influence human systems, and the genetics and engineering required to mass produce them.
So we're essentially mapping out the ultimate chemical arms race.
We are an arms race between humanity and the microbial world, all detailed in our source material.
And the aha moment for why this all matters, it really comes right in the definition itself. When we talk about secondary metabolites, we aren't talking about the necessary stuff.
No, not at all.
We're not talking about the chemical machinery that lets an organism breathe or replicate its DNA or, generate basic energy. We're talking about these specialized, often unique chemical compounds that are not strictly essential for its basic life functions.
Exactly. And that's the fundamental difference to grasp. You have primary metabolites, things like citric acid, ethanol, your standard amino acids, and they are ubiquitous.
Every organism needs them.
Every organism. They're produced through major common metabolic pathways, and they're typically made during the rapid exponential growth phase.
And then on the other side, you have the secondary metabolites.
These are the specialized tools. They're only produced by specific groups of organisms. They have these complex and often novel chemical structures. And critically, they're usually only produced when the culture is stressed out.
Or when it's entered the stationary phase.
Right, after all that rapid growth is complete. It's a clear transition from a growth strategy to, really, a survival strategy.
And yet, these non-essential chemicals include, as our source material points out right away, the drug that transformed the 20th century, penicillin.
The big one.
But it's not just penicillin. As we'll see, this group also includes the core drugs we're using right now to fight cancer and regulate high cholesterol.
Yeah.
It is one of the most commercially and medically vital areas of biotechnology today.
Without a doubt.
So let's start there. Let's unpack that foundational story of penicillin. For decades, the narrative was always the same. Alexander Fleming, 1928, an untidy lab, a contaminating penicillium mold, and just pure, unadulterated luck.
It's the perfect scientific fairy tale.
It is, but it's also
Deeply misleading. It completely strips away the context of Fleming's career. This was a man who was dedicated to finding agents that could lyse bacterial cells, things that could be used therapeutically.
This wasn't his first rodeo, so to speak.
Not at all. Before penicillin, he had already discovered the enzyme lysozyme. And that discovery, it showed his specific research focus. But he was ultimately disappointed because lysozyme didn't work against many of the common, dangerous human pathogens.
Okay, so when he noticed that penicillium mold inhibiting bacterial growth, it wasn't some random occurrence to him. It was the continuation of a focused, career-long quest.
Absolutely. The real genius wasn't in just spotting the mold. I mean, dozens of scientists had observed similar inhibition phenomena before him and just dismissed it.
They just cleaned the plate and moved on.
They did. Fleming's critical contribution was isolating the active substance, naming it, and most importantly, testing it therapeutically in an animal model. He was the one who showed that the culture filtrate had actual clinical efficacy, even if the early yields were, incredibly low.
And the industrial breakthrough, the massive, large-scale production and purification needed for the clinic, that came much later.
Much later. That was a massive collaborative effort, spurred primarily by the needs of World War II, more than a decade after his initial discovery.
This historical context, it sort of leads to a crucial philosophical shift about what these chemicals are for. We call them natural antibiotics weapons designed solely to kill competitors. But is that assumption really holding up today?
It's a bit shaky. I mean, the idea that every secondary metabolite is just a biological weapon is too simplistic. For one thing, many of these compounds, even the classic ones, show only marginal antimicrobial activity in a standard lab assay.
So they're not that great at killing things on their own?
Not always. They often require chemical modification, the creation of what we call semisynthetic agents, to become truly potent therapeutic drugs. And secondly, many are produced by the source organism at extremely low, almost undetectable concentrations.
So if they aren't weapons, what's their function in nature?
Well, the prevailing hypothesis now is that many of these compounds primarily function as signaling molecules.
Signal.
Yes, they allow organisms to communicate with each other, perhaps to sense high cell density, what we now call quorum sensing, or to indicate a severe resource limitation. This fits really well with the observation that a large number of secondary metabolites show no antimicrobial activity whatsoever.
So the original purpose might be communication, not conquest.
It seems that way for many of them. However, we can't completely dismiss the original antibiotic role. There's strong evidence suggesting that some compounds, like penicillin, did function as natural antibiotics in their native environments.
And how do we know that?
We know because the genes that code for resistance against beta-lactams, the enzymes that destroy penicillin, are frequently found not just on those mobile resistance plasmids, but embedded right on the chromosomes of many bacterial species.
Ah, so that implies a long evolutionary history with the threat.
A very long history. It suggests these bacteria encounter and genetically adapted to these natural chemical threats long before humans ever synthesized the first dose.
Okay, so the moment the pharmaceutical industry recognized that not all these culture filtrates were just bacterial weapons, they initiated a massive screening shift.
They're a complete pivot.
They stopped focusing solely on killing bugs and started asking a much broader question.
And that was the real turning point in the field. They started screening these culture filtrates for agents that could, say, inhibit specific animal enzymes or bind to crucial receptors on animal cell surfaces.
And that opened up the market for treating non-infectious diseases.
Everything. Cancer, high cholesterol, blood pressure, immunosuppression, all using microbial compounds.
So let's start with one of the biggest, the antitumor agents. It's kind of ironic that so many of our most important cancer fighters come from the same source as traditional antibiotics, the Streptomyces species.
It is, and the common mechanism here is that they all target the very core of the cell, the DNA. We see several just brilliant chemical strategies. Take the anthracycline family, which includes drugs like doxorubicin and doxorubicin. They use a tactic called intercalation.
Intercalation, what does that mean exactly?
It means they physically insert their flat chemical structure. These compounds have these fused benzene rings right in between the base pairs of the DNA double helix.
Like slipping a card into a deck.
Exactly like that. And this physical insertion dramatically changes the shape of the DNA, which prevents crucial enzymes like topoisomerase from doing their job of unwinding and managing the DNA during replication or transcription. It effectively halts cell division.
Which is why they're so effective against rapidly dividing cancer cells.
Precisely. We see a similar strategy with another drug, dactinomycin, which also intercalates, stopping both DNA transcription and synthesis. But then you have a structurally different approach with agents like bleomycin.
How does that one work?
Bleomycin is a chemical radical generator. It complexes with a ferrous ion and then generates oxygen-free radicals. Because its structure allows it to bind right near the DNA, those highly reactive free radicals are generated right next to the target.
So using direct damage.
Active destruction. It literally cleaves the DNA strands.
And then there's the most complex example, xynostatin, or neocarzinostatin, which sounds like a chemical payload wrapped in a protective protein cloak.
It's an absolutely ingenious delivery system. The core is the chromophore, which is the active part, but it's protected by an apoprotein, and the mechanism requires activation.
It has to be armed inside the cell.
Exactly. When the chromophore is released from that protective protein and then reduced, usually by something like glutathione, which is present in the cell, it becomes a free radical. And since the molecule's naphthalene ring system also intercalates into the DNA, that radical is perfectly positioned to oxidize the DNA backbone,
Causing a break.
causing precise, targeted strand cleavage. It really is like a guided missile carrying a tiny bomb.
That's incredible. Okay, so moving away from DNA targets, a huge success story came from targeting specific enzymes, particularly protease and peptidase inhibitors. This field was essentially founded by Hamao Umezawa in the late 1960s.
Umezawa's insight was recognizing that proteases, these are the enzymes that cleave protein chains, are central to all sorts of pathological processes.
Like what?
Well, viral polyprotein processing, like the AIDS virus protease, and even basic physiological events like blood clotting. So screening culture filtrates for protease inhibition yielded these highly valuable research regions like anti-pain and pepstatin, but also therapeutics like B-statin, which is used for immunopotentiation in cancer patients.
And that same screening shift led to maybe one of the most profitable discoveries for non-infectious disease,
Yeah.
the search for AC inhibitors.
Crucial for blood pressure control, they isolated a compound called A58365A, a streptomyces product, which potently inhibits angiotensin-converting enzyme, or ACE.
And ACEs is a key component of the system that regulates blood pressure.
It is, but what's so amazing about this specific molecule is how it relates to rational drug design.
Because scientists had already been working on synthetic ACE inhibitors, right?
Mm-hmm.
Starting from snake venom peptides and simplifying them down to compounds like captopril.
Exactly. And the natural microbial product, A58365A, turned out to be what's called a conformationally restricted analog of one of the synthetic intermediates that chemists had rationally conceived of.
So nature had already built a better version of what they were trying to make.
In a way, yes. Nature had already optimized the structure for binding and stability, proving that the microbial world can offer these chemical scaffolds for novel structures that we just couldn't have designed from scratch.
That exact principle seems to be reinforced by the history of the statins, the drugs that target high cholesterol.
The statin story is a perfect example. It began in the 1970s when scientists isolated compounds from penicillium species that successfully inhibited an enzyme called HMG-CoA reductase.
And that enzyme catalyzes the first unique and therefore the rate-limiting step of cholesterol synthesis in the liver.
That's the one. They found a compound called compactin, and compactin worked by acting as a competitive inhibitor of HMG-CoA reductase. Its structure looked so much like the natural substrate that it just clogged up the enzyme's active site.
And that led to lovastatin.
It did. Merck later isolated mivinylin from Acebillus, which became marketed as lovastatin. The success of these microbial discoveries ultimately spawned an entire generation of synthetic statins, like atorvastatin or Lipitor, leading to billions in global sales. This single discovery changed the trajectory of cardiovascular health worldwide.
Before we move on, we have to mention the critical class of immunosuppressants, which are vital for organ transplantation, and they were also found through this non-traditional screening.
The key example here is cyclosporin A, a cyclic peptide isolated from a fungus. And what's fascinating is that it was originally screened for its antifungal properties.
So it was a complete accident.
A happy accident. Its discovery shows how serendipitous this kind of non-target screening can be.
So walk us through its mechanism. It's highly specific, I understand.
Incredibly specific. Cyclosporin A doesn't directly target the immune system. Instead, it forms a complex with a cellular protein called cyclophilin. This new complex then binds to and inhibits another enzyme, calcineurin, which is a key phosphatase.
So it's a multi-step process.
It is. By stopping calcineurin, the drug prevents a necessary dephosphorylation step for a transcription factor to enter the nucleus, which ultimately blocks the activation and proliferation of T cells. It shuts down the immune rejection response at a really fundamental genetic level.
And other drugs like rapamycin and FK506 followed this same path. It really demonstrates that the microbial world just contains the blueprints for regulating almost any physiological process in a higher organism.
Absolutely. Even compounds that simply bind to receptors like asparagine from Aspergillus, it binds to the cholecystokinin receptor, a hormone involved in digestion. And just like the ACE inhibitor, asparagina's complex, unique structure provided a chemical scaffold that no chemist would have ever conceived of when trying to design an analog of the native peptide hormone.
Okay, let's pivot back to the traditional arms race, the antibacterials.
Yeah.
Before we talk about mechanisms, we have to set the stage by defining the great wall that these drugs have to cross, the gram-negative outer membrane.
This barrier is everything. It dictates which drugs work and which ones simply don't. Gram-negative bacteria have two membranes, and that outer one acts as a formidable selective barrier.
So how do things get through?
Small, hydrophilic sleuths, typically less than 1,000 Daltons, can cross through these water-filled channels called porins.
But the porins aren't just wide open doors.
No, not at all. In E. coli, the channels are very narrow, meaning larger water-soluble agents really struggle to diffuse through. And crucially, the membrane itself is structurally unique. The outer leaflet is composed of lipopolynitrite, or LPS.
And that has a big effect on permeability.
A huge effect. Because of the LPS, the membrane has unusually low permeability to lipophilic or fat-loving molecules, which normally cross other membranes quite easily.
So the predictable takeaway here is that larger and more hydrophobic antibiotics are usually restricted to fighting gram-positive bacteria, which of course lack this outer barrier entirely.
That's our baseline challenge. Now, with that in mind, let's introduce the cornerstone of antibacterial warfare. The beta-lactams, starting with penicillin G. Their primary target is the unique structural element of bacteria. the cell wall made of peptidoglycan.
And how does that target actually work? What is peptidoglycan?
Peptidoglycan is a giant interlocking network. You can think of it as long polysaccharide chains alternating N-acetylglucosamine and N-acetylmuramic acid residues, which are then cross-linked by short peptides.
And that cross-linking is the key.
It's essential. It's what gives the cell wall its massive mechanical strength, allowing the bacterial cell to resist the huge internal osmotic pressure. If you stop that cross-linking, the cell just bursts.
And the enzyme that does the cross-linking is DD transpeptidase. So how does penicillin shut it down?
This is where the structural brilliance of the beta-lactam ring comes in. The ring system structurally resembles the D-alanine portion of the natural peptidoglycan substrate.
So it's a mimic.
It's a perfect mimic. So when the DD transpeptidase enzyme attempts to perform its normal function cleaving, that D-allyl bond and forming a cross-link, it encounters penicillin instead.
It's a chemical trap.
Precisely. Penicillin interacts with the enzyme, but instead of completing the reaction, the beta-lactam ring opens up and forms a covalent bond with the active site serine residue of the enzyme. This forms a stable, permanent penicilloyl enzyme complex.
Which irreversibly inactivates the transpeptidase.
Completely inactivates it. This mechanism makes penicillin a suicide inhibitor.
A suicide inhibitor.
That's right. It mimics the substrate, it tricks the enzyme into acting on it, and in the process, it permanently destroys the enzyme's function. This irreversible inhibition is far superior to a simple competitive inhibitor because it guarantees complete target shutdown, which is almost always lethal to the cell.
But despite this elegant, lethal mechanism, classic penicillin G still struggles against most gram-negatives.
And that comes right back to that outer membrane barrier. Due to its lipophilicity, it just can't cross efficiently. And that single failure point drove the next several decades of microbial drug development, as chemists desperately tried to find ways to make this core mechanism work against gram-negative threats.
So the microbial world didn't put all its eggs in the beta-lactam basket. Let's look at the other major classes that were discovered, many of which target the second essential bacterial process, protein synthesis. We can start with the macrolides.
The macrolides are complex structures defined by a large lactone ring, like erythromycin. They're products of the polyketide biosynthesis pathway, which we'll get into later, and they come from Streptomyces relative.
And they have the same gram-negative problem.
They do. Because they are large and hydrophobic, they primarily target gram-positive bacteria. We need chemical modifications, semisynthetic derivatives like azithromycin, to push their activity even slightly into the gram-negative domain.
Then you have the antimycins, like rifamycin SV, which target the control room of the cell, RNA synthesis.
The antimycins inhibit prokaryotic RNA polymerase, and again, naturally large and hydrophobic, so their activity is mostly gram-positive. But the modification that created rifampicin, just adding a polar group, gave it better penetration and, critically, activity against tough pathogens like Mycobacterium tuberculosis.
So it became a mainstay in treating TB.
A crucial one.
Now, we find the first truly successful broad-spectrum microbial products that are not modified beta-lactams, the tetracyclines. What makes them broad-spectrum where penicillin G failed so badly?
The tetracyclines are a fused four-ring system, also from Streptomyces. They inhibit prokaryotic protein synthesis. Their secret weapon is their structure. They are relatively hydrophilic due to all their hydroxyl, amide, and avisamine groups.
And that hydrophilicity is the key.
It's the key. It allows them to efficiently traverse the gram-negative outer membrane via those porin channels. This means they can reach the target ribosomes inside both gram-positive and gram-negative cells.
This drug was used everywhere historically, which ended up creating some problems.
It certainly did. The widespread use of tetracyclines, including their long-standing use as animal feed additives for growth promotion, selected for and disseminated resistance plasmids in pathogens like Salmonella. It was a really potent lesson in the immediate feedback loop of the arms race. Pressure leads to resistance.
And the same broad-spectrum principle applies to chloramphenicol, another small molecule protein synthesis inhibitor.
Chloramphenicol is so small and simple that it's now cost-effectively produced chemically rather than relying on the microbial source, Streptomyces venezuelae. Its small size allows it to easily penetrate gram-negative porins.
But it has a major caveat.
It does. Toxicity. It can inhibit mitochondrial protein synthesis in our own eukaryotic cells, leading to serious side effects like bone marrow suppression. Its use today is highly restricted, often reserved for cases like typhoid fever, where the pathogen hides inside human cells.
Finally, we have the specialized peptide antibiotics from Bacillus species. Most are highly toxic and used topically, but we need to discuss the radical exception, polymyxin.
Polymyxin is a really unique tool in the gram-negative fight. It's polycasonic, and it targets the outer membrane directly. It binds strongly to the anionic LPS in the gram-negative outer membrane, disrupting its entire molecular structure.
So it just rips the membrane apart.
Pretty much. Once that structural integrity is gone, the hydrophobic tail of polymyxin binds and inserts into the cytoplasmic membrane, causing permeabilization and cell death. It's a brute force weapon used today against highly resistant gram-negative organisms like Pseudomonas aeruginosa.
And before we move on, we can set up our next deep dive. The aminoglycosides. They're also protein synthesis inhibitors, but they have that unique characteristic of being equally effective against both gram-positive and gram-negative bacteria.
Right, due to their hydrophilicity and size. We'll definitely return to their story of resistance and rational design shortly.
We've established that the gram-negative cell wall is a barrier, but our source material makes it really clear that fighting fungi presents an entirely different and in some ways much harder problem.
The difficulty is rooted in the fact that fungi are eukaryotes, just like we are. They share our cellular machinery for protein and nucleic acid synthesis. This vastly limits our ability to find safe, selective targets.
So to be effective and have low toxicity, antifungals have to hit structures or pathways that are totally unique to the fungal cell.
Exactly. For instance, there's polyoxin B, which is used in agriculture. It's effective because it mimics a precursor molecule and inhibits the synthesis of chitin, which is a unique component of the fungal cell wall. Another drug, grizofulvin, inhibits mitosis by binding to tubulin proteins.
And the toxicity problem is perfectly illustrated by the polyenes.
The polyenes, like philippin, are macrocyclic lactams that attack the cell membrane by complexing with sterols. By binding to the sterols, they increase the membrane's permeability, effectively punching holes in the cell.
But the problem is we have sterols too.
We do. The high toxicity comes because our animal cells contain cholesterol, which is a type of sterol, while fungal membranes contain ergosterol, which is the primary target. The cross-reactivity with our cholesterol is enough to limit polyenes mostly to topical applications.
But new classes like the echinocandins offer better selectivity.
They do, by focusing on other unique components of the fungal cell wall polysaccharide synthesis, making them essential tools for systemic fungal infections.
So now let's look at the big picture goals driving research in this field today. The arms race is clearly the central narrative.
The main focus is, and has to be, the development of new agents. This is an endless task driven by two things. First, the lack of effective, low-toxicity agents for inherently difficult pathogens like fungi, viruses, and intrinsically resistant bacteria like P. aeruginosa.
And the second driver.
The second point is the overwhelming driver, the relentless, accelerating emergence of resistance, which severely limits the useful lifespan of any new drug we discover.
And this situation is especially dangerous in hospitals.
Hospitals are the perfect breeding ground. They concentrate massive amounts of antibiotics, which rapidly selects for resistant bacteria, leading to dangerous nosocomial or hospital-acquired infections among vulnerable patients who already have compromised immune systems. This constant evolutionary pressure is what turns drug discovery from a scientific pursuit into an urgent, ongoing military campaign.
Okay, so since we are fighting an arms race, let's zoom in on a specific battlefield, the development of the aminoglycosides. Because this class just perfectly captures that back and forth between discovery, microbial defense, and our targeted chemical counterattacks.
It's a fantastic case study. The core of the aminoglycosides is the aminocyclitol moiety, that ring structure, either streptidine or 2-deoxystreptamine, which is substituted with amino sugars. And the key characteristic is that they are polycationic.
Meaning they have multiple positive charges.
Multiple positive charges, which is absolutely vital for both their function and their toxicity.
And, as we noted, they are unique among protein synthesis inhibitors because they are bactericidal. They kill the cell irreversibly, unlike the bacteriostatic action of tetracyclines or chloramphenicol. Why is that?
That's explained by this powerful cascading mechanism known as the Davis hypothesis, which describes an irreversible entry process. It's a brilliant sequence of events.
Walk us through it.
Okay, step 1. A tiny amount of the polycationic drug manages to enter the cell, and because of its positive charge, it binds very strongly to the anionic 16S rRNA within the bacterial 7DS ribosome.
So it finds its target.
It finds its target. Step 2. This binding doesn't just block synthesis, it physically damages the ribosome, causing gross misreading and truncation of the polypeptides being made.
So it's making junk proteins.
Total junk proteins. Step 3. These new, faulty, often hydrophobic polypeptides insert themselves into the bacterial membrane, making it leaky.
Ah, I see where this is going.
And this is the kill shot. Step 4. The cell's interior is negatively charged. The now leaky membrane allows that strong interior negative membrane potential to suck in massive lethal amounts of the polycationic drug. This huge influx causes a complete cessation of protein synthesis and guaranteed cell death. It is literally a self-fulfilling prophecy of destruction.
That is an elegant trap. But focusing on the misreading part, how does the drug actually cause the ribosome to misread the genetic code?
Structural studies have revealed the precise molecular mechanism. Protein synthesis is usually highly accurate because the codon and anticodon pairs in the ribosomal A site have to interact with specific bases of the 16S RNA to stabilize the complex. If there's a mismatch, the complex just falls apart.
It's a proofreading step.
It is. But aminoglycosides bind right near these critical 16S RNA bases, and they force the structure of the 16S RNA into the correct conformation, essentially stabilizing the binding even when there is a mismatch. They turn a proofreading step into a blind acceptance step, which drastically increases the misreading frequency.
Okay, so now for the microbial counterattack. Aminoglycoside resistance. Our source material is really adamant that while we can make ribosomal mutants in the lab, clinical resistance is almost entirely due to R plasmids, these mobile genetic elements that carry drug inactivation enzymes.
The bacterial defense strategy is straightforward. Destroy the positive charge. The 3 major enzyme families carried on these R plasmids, the AACs, acetyltransferases, the APHs, phosphoryltransferases, and the ANTs, nucleotidyltransferases, all work by enzymatically attaching groups to the antibiotic.
So they modify the drug itself.
They do. Attaching acetyl groups removes a positive charge. Attaching phosphoryl or nucleotidyl groups adds a negative charge. In any case, this negative or neutral modification prevents the drug from binding to the anionic 16S RNA, or prevents its critical charge-driven entry into the cell.
We also have to address the unfortunate limitation of this class, toxicity. Since they are polycationic, they are somewhat toxic to human tissues, causing inner ear and kidney damage.
Right, which means their use is strictly limited to life-threatening systemic infections, particularly against difficult gram-negative bacteria like P. aeruginosa. We use them because they provide a systemic advantage in that gram-negative war. They can cross low-permeability outer membranes by binding to the anionic surfaces of the bacteria and disorganizing the barrier. But once resistance emerges, we lose that critical advantage, forcing us to find new solutions.
And the story of fighting back against resistance starts with kanamycin in 1957.
Kanamycin was a natural discovery that lacked a crucial structural element, the 3'' hydroxyl group. And this group was the primary target of the main streptomycin inactivating enzyme, APH3-NARG. So simply by changing the aminocyclidyl core, kanamycin was instantly active against many streptomycin-resistant strains.
Which showed chemists where the vulnerability was.
Exactly. And this paved the way for the rational design era led by chemists like Sumio Umezawa.
What was his approach?
Umezawa took that structural knowledge that APH3 was phosphorylating a hydroxyl group and rationally designed a fix. He chemically removed the targeted hydroxyl groups at both the 3' and 4' positions of kanamycin, creating dabecacin. It was a drug built purely to defeat the known resistance mechanisms, and it became extremely popular in Japan.
But the truly revolutionary drug in this family, amikacin, was inspired by a natural chemical feature found in a completely different organism.
Yes, this is a great story. Scientists noted that butyrosine A from a bacillus species had this bulky 4-amino-2-hydroxybutyryl substituent on its 1 amino group. So they tried adding this exact bulky substituent to kanamycin A.
And that created amikacin.
It did, and the bulky group acted like a molecular shield. It abolished modification at the 2-hydroxyl group due to steric hindrance. It physically blocked the enzyme from being able to access that site.
And it also protected other sites, too.
It did, which was a remarkable and somewhat unpredicted result. It also conferred resistance to enzymes modifying the 3-amino group of 2-deoxystreptamine. The hypothesis here is skewed binding. The bulky substituent shifts the drug's orientation in the enzyme's active site, moving the modification sites away from the catalytic center, preventing destruction.
So amikacin became a key second-line drug.
A huge one, a testament to combining natural inspiration with rational modification.
And it turns out nature was doing this job all along. Tobramycin and gentamicin C are perfect examples of natural products that already had the resistance-evading structures that chemists later fought so hard to invent.
It's true. Tobramycin naturally lacked the 3-phet-hydroxyl group, proving that natural evolution had already achieved the resistance mechanism chemists were seeking. And similarly, gentamicin C naturally lacked both the 3-ham-4-phet-hydroxyl groups.
And let's quickly touch on mutasynthesis, this idea of tricking the microbial factory into making novel drugs for us.
Mutasynthesis is an ingenious use of genetics. You create a mutant strain that is blocked in the synthesis of a natural intermediate. Then you feed that mutant a synthetic analog of that intermediate, exploiting the broad substrate specificity of the biosynthetic enzymes.
You're swapping out a part on the assembly line.
Exactly. For example, feeding a specific synthetic neamine analog to a bacillus mutant resulted in a butyrosine analog that was resistant to two major modifying enzymes simultaneously. It's targeted combinatorial chemistry done by the microbe itself.
And the resistance genes themselves, where did they come from in the first place?
Julian Davies proposed that they originated in the antibiotic-producing organisms themselves as a necessary mechanism for self-protection.
That makes sense.
And this was confirmed by cloning, which showed the R plasmid genes are highly homologous to genes found in producing actinomycetes. Furthermore, the modifying enzymes evolved from common primary metabolism enzymes. It looks like APHs, the phosphoryltransferases, resemble protein kinases, and AACs, the acetyltransferases, resemble protein acetylases.
Which brings us to a crucial and pretty sobering takeaway, illustrated by the geographical data. Antibiotic usage patterns directly shape the resistance profiles we see around the globe.
This is critical for global health policy. In countries that heavily used kanamycin and its derivatives, the most prevalent resistance mechanism was AAC6, which inactivates those specific drugs. Conversely, in the U.S., where gentamicin was historically favored, the most common enzymes were ANT2 and AAC3, which efficiently inactivate gentamicin.
So the microbe adapts precisely to the selective pressure we apply.
Exactly. The geographic map of resistance is a direct mirror of our drug usage.
If the aminoglycosides show the power of rational design, the beta-lactams really show the never-ending escalation of the arms race, especially given their massive commercial importance and incredibly low toxicity to humans.
Their low toxicity is due entirely to their target peptidoglycan synthesis, which is unique to bacteria, and penicillin G production was quickly maximized. Crucially, the fermentation process required precise control over precursors.
Right, adding phenylacetic acid gave you penicillin G.
And adding phenoxyacetic acid yielded penicillin V, which was acid-resistant and could be taken orally. But the natural compounds had those two major flaws, staphylococcal resistance and gram-negative ineffectiveness. This spurred the semisynthetic revolution.
To create thousands of new compounds.
Exactly. Scientists needed the core structure, the 6-aminopenicillinic acid or 6-APA nucleus. They eventually found an enzyme, penicillin acylase, which could cleave the side chain off penicillin G, leaving the beta-lactam ring intact and ready for chemical modification. Every major beta-lactam used today is derived from this semi-synthetic process.
So let's follow the first counterattack, fighting staphylococcal penicillinase, the class A beta-lactamase.
The strategy was steric hindrance. The goal was to attach bulky substituents on the alpha carbon of the 6-substituent to physically block the enzyme from accessing the reactive beta-lactam ring.
And that gave us methicillin and naphthalene.
It did. They are incredibly stable against the staphylococcal enzyme, and they bought us decades of efficacy against S. aureus, before, of course, the emergence of MRSA.
The second counterattack was gaining gram-negative activity. Penicillin G was too lipophilic to cross the outer membrane. The solution was ampicillin.
By introducing a simple amino group on the alpha carbon of the benzyl substituent, they created ampicillin. This one addition changed the molecule from anionic and hydrophobic to zwitterionic and more hydrophilic. Suddenly, ampicillin could diffuse across the gram-negative outer membrane much faster via the porin channels.
But gram-negatives have that two-barrier defense, the outer membrane plus the periplasmic beta-lactamase enzyme. Ampicillin has to contend with both.
That's right. And ampicillin works because it's a synergistic advantage. It crosses the outer membrane faster, and it's generally more resistant to the gram-negative beta-lactamase than penicillin G is. This allows a sufficient concentration to reach the target DD transpeptidase.
And this approach was refined for even tougher bugs like P. aeruginosa.
It was, with drugs like carbapenems and 2-carcillin, which added negatively charged groups that enhanced activity and were weaker inducers of the chromosomal beta-lactamase.
This brings us to the cephalosporins, which feature a 6-member dihydrothiazine ring fused to the beta-lactam. The natural product, cephalosporin C, already had low gram-negative activity and penicillinase resistance, a potent combination from the start.
The tricky part was creating the core starting material, 7-aminocephalosporinic acid. Unlike penicillin, they couldn't find a natural enzyme to cleave the side chain. It took chemical methods, a ring expansion from penicillin, and later a 2-step enzymatic process to make semisynthesis viable.
The first-generation cephalosporins solved the dual activity problem, but created a paradox when looking at the gram-negative resistance enzymes.
This is a crucial teaching moment about enzyme kinetics. When studying the chromosomal beta-lactamases in gram-negatives, scientists initially called them cephalosporinases because, at extremely high substrate concentrations, the enzyme appeared to hydrolyze cephalosporins faster than penicillin G. This was a measure of maximum speed, or Vmax.
But that failed to predict how well the drug worked in a patient.
Correct. When you look at low, clinically relevant concentrations, the true measure of efficiency is Vmax divided by demolars, which incorporates the enzyme's affinity. And measured this way, the chromosomal enzyme actually hydrolyzes penicillin G 72 times faster than cefazolin.
So the efficacy of the first-gen cephalosporins relies on their superior ability to penetrate the porins and their low affinity for those chromosomal beta-lactamases.
That's the key insight.
But as soon as these drugs became widespread, the R plasmids, carrying the broad-spectrum TEM beta-lactamase, emerged in gram-negatives. The microbes learned to fight back quickly,
which led to the second-generation cephomycins. Scientists screened phylogenetically distant streptomyces and found cephomycins, which possessed a unique methoxy group at the 7-alpha position. This subtle chemical feature made them absolutely resistant to that widespread TEM enzyme.
But this led to a new resistance problem: intrinsically resistant organisms like E. cloacae and P. aeruginosa. This required the third-generation cephalosporins.
These drugs, like cefotaxime and ceftazidime, were synthetic breakthroughs, featuring a substituted oxime group. They performed miraculously against organisms that resisted the cephomycins. And the reason comes back to induction.
Induction of the enzyme.
Right. The cephomycins were strong inducers of the chromosomal beta-lactamase, causing the organism to pump out massive amounts of the enzyme when exposed. The third-generation compounds, by contrast, had almost no inducer activity, which kept enzyme levels low and allowed the drug to work effectively.
So the bacteria responded, of course, by producing constitutive mutant strains that permanently produced the chromosomal beta-lactamase at high levels, regardless of induction.
The arms race continues, and this required the fourth-generation cephalosporins, like cefepime. They retained the resistance groups, but added a quaternary nitrogen atom. Cefime was engineered to have a vastly lower affinity, a lower anoles for the chromosomal enzymes than the third-gen drugs.
So even with high enzyme levels, the enzyme couldn't grab the drug.
Exactly. Even though the constitutive mutants were pumping out high levels of the enzyme simply couldn't grab onto cefepime efficiently enough to destroy it before it reached the peptidoglycan target.
Because resistance is so relentless, especially with new TEM mutants evolving to destroy even the third and fourth generation cephalosporins, researchers had to look for structures that completely departed from the penicillin and cephalosporin nuclei.
This led to non-traditional screening methods, often looking for agents that didn't kill bacteria outright, but, say, inhibited crucial bacterial enzymes. This brought us to the carbapenems.
These are radical structures, like phenamycin, where a carbon replaces the sulfur in the penicillin nucleus, plus there's a double bond.
Its semisynthetic derivative, imipenem, is extremely broad spectrum. It is so effective because it acts as a suicide inhibitor against the class C chromosomal beta-lactamases, the very ones that cause problems with the third-generation drugs.
But imipenem has a weakness.
It does. Its activity against P. aeruginosa comes from rapid penetration through a specific outer membrane channel used for amino acids. A single mutation can cause the loss of this one channel, leading to rapid high-level resistance.
The other key departure is the drug enhancer, the clavams, like clavulanic acid.
Clavulanic acid was isolated by specifically screening for beta-lactamase inhibitors, not antibiotics. It contains an oxygen instead of sulfur and has almost no antibiotic activity itself.
But it's a protector.
It's a bodyguard. It is an efficient, irreversible suicide inhibitor of the class A beta-lactamases, like the staphylococcal and TEM enzymes. It's never administered alone. It's sold with amoxicillin as augmentin, sacrificing itself to protect the partner drug.
And finally, we have the monobactams, featuring an isolated, non-fused beta-lactam ring.
Interestingly, the natural monobactams were derived from gram-negative rods, not the traditional streptomyces or fungi.
None.
While they had low initial activity, chemists used their structure to synthesize aztreonam, which is highly effective against a broad range of gram-negative bacteria.
To bring this section full circle, rational design is now highly informed by structural biology. When you compare the structure of a beta-lactam to the target enzyme, the PBP, what's the core difference that explains the outcome?
It's the recycling mechanism. Structural studies show the beta-lactam and the PBP have a remarkably similar overall fold. Both enzymes are acylated by the beta-lactam at an active site serine.
But one gets stuck and the other doesn't.
Exactly. The class O beta-lactamase contains a key feature, an activated water molecule, often facilitated by a glutamic acid residue. This activated water rapidly hydrolyzes the complex, regenerating the enzyme so it can go kill another drug molecule. PBPs lack this activated water, thus they remain permanently inactivated by the drug.
So that understanding is key. Future drugs must be designed to bind to the PBT structure while avoiding that specific cavity where the beta-lactamase activates its recycling water molecule.
That's the holy grail of rational design in this field.
It's one thing to discover these complex molecules, but another entirely to produce them at the industrial scale required for global medicine. The traditional method for yield enhancement sounds incredibly rough.
It was a shotgun approach. Heavy random mutagenesis using agents like X-ray or UV light, followed by laborious screening for high producers. The process was slow, but it was incredibly effective.
Just how effective?
Fleming's original penicillin strain produced about 3 milligrams per liter. Over decades of traditional mutagenesis, yields rose to 7,000 milligrams per liter. Today, they are over 40 grams per liter.
That's an astonishing increase, but the output went up while the strains themselves were often weakened.
Exactly. Random mutagenesis accumulated all these unwanted mutations that weakened the organism's vigor. So classical genetics had to be used to fix this. Techniques like the parasexual cycle in fungi or protoplast fusion in streptomyces allowed scientists to backcross the high-producing strains with hardy wild-type parents to restore growth vigor while retaining the high-yield traits.
And modern genomics later revealed the genetic secret behind these massive yield improvements.
It did. Sequencing streptomyces genomes confirmed they are massive, almost twice the size of E. coli's genome, and they have incredibly complex regulatory systems. But crucially, sequencing confirmed that antibiotic genes are typically grouped together in tight clusters.
And in the high-producing strains.
When they analyzed the high-producing P. chrysogenum strains created by those traditional methods, they found the secret. The strains had up to 50 copies of the penicillin biosynthesis genes amplified directly on the chromosome. The organism had literally duplicated its way to high production.
This genomic knowledge opened the door to targeted engineering. Let's talk about the incredible complexity of the polyketide biosynthesis pathway, which makes macrolides and tetracyclines.
This is truly nature's Lego set. Polypeptide biosynthesis is a modification of fatty acid synthesis, but it uses these massive multifunctional enzymes called polypeptide syntheses, or PKS. The revolutionary insight is the type I modular system.
Okay, how does that modular system work?
The synthesis is carried out by giant proteins that are divided into a sequence of modules. Each module is responsible for adding one building block, say a C3 propionate unit, to the growing chain. And within that specific module, there are specific enzyme domains, like a reductase or a dehydratase, that determine the precise chemical structure at that step.
So the final structure is dictated solely by the sequence and composition of those modules.
That's it, which means scientists can now use genetic modification to swap modules between different gene clusters. This allows for the rapid, targeted creation of hundreds of novel hybrid antibiotics, what we call combinatorial biosynthesis.
And the other major pathway is non-ribosomal peptide synthesis for drugs like beta-lactams and cyclosporin A.
This pathway is also modular. Each module activates and adds a single amino acid. This non-ribosomal mechanism is what explains how the producers can incorporate unnatural amino acids, like D-amino acids, which standard protein synthesis can't handle.
And there's an interesting evolutionary footnote here.
There is. The genes for beta-lactam biosynthesis are strongly homologous between bacteria and fungi, which suggests a relatively recent horizontal gene transfer event across kingdoms.
And finally, genetic engineering is used to break bottlenecks to increase yield.
Absolutely. The best example is the cephalosporin producer, A. chrysogenum. Researchers identified the conversion step from penicillin N to cephalosporin catalyzed by the expandase gene as a bottleneck. By simply introducing an extra copy of this cloned gene, they increased cephalosporin yield by 20% to 40% and drastically decreased the secretion of the unwanted precursor.
We established early on that secondary metabolism is tied to stress or limited growth. Let's explore the regulatory control mechanisms that determine when these industrial powerhouses decide to start synthesizing their valuable products.
The primary rule is that antibiotic production usually only occurs when cultures are entering or are already in the stationary phase. This is why continuous fermentation, which is great for primary metabolites, is often unsuitable for antibiotics. The cell has to finish its rapid growth before it diverts energy to survival chemistry.
And the signal for this transition often involves intercellular signaling or communication between the cells.
Correct. Both antibiotic production and sporulation in organisms like Streptomyces are modulated by signals, including peptides and membrane-permeable lactones. These lactones are chemically similar to the quorum-sensing signals used by gram-negative bacteria.
And they accumulate at high cell density.
Right, acting as a chemical link between the slowdown of growth and the initiation of the secondary metabolism genes.
We also see familiar repression mechanisms affecting the yield, which means you need precise nutrient control in the fermenter.
Catabolite repression is very common. Excess carbon sources like glucose will decrease productivity, so they have to be fed carefully and incrementally. Excess nitrogen or phosphate also severely decreases production, likely by inhibiting gene transcription. And there is also evidence of feedback regulation, where the antibiotics themselves exert negative feedback, signaling the cell to stop production if levels get too high.
Now, let's discuss the single best anecdote that illustrates the deep physiological differences between these microbial factories, the lysine paradox. This involves the essential precursor for beta-lactams, alpha-aminoadipic acid.
This is a perfect example of why you can't use the same fermentation strategy across different organisms. In fungi, like Penicillium chrysotologinum, alpha-aminoadipic acid is an intermediate on the pathway to synthesize lysine.
So if you add excess lysine to the medium,
The fungus senses that it has enough, and feedback inhibition shuts down the entire pathway. The result? A shortage of alpha-aminoadipic acid, which strongly inhibits penicillin production.
but if you apply that same logic to a prokaryote producer, you get the exact opposite result.
Precisely. In prokaryotes, like Streptomyces, alpha-aminoadipic acid is synthesized from lysine. Therefore, adding excess lysine efficiently converts into the desired precursor, alpha-aminoadipic acid, and actually stimulates cephamycin production. Understanding these core metabolic differences is absolutely vital for designing the right feeding schedule.
So this strict regulatory control dictates that industrial antibiotic production has to be a two-stage process.
That's right. Stage 1. Initial submerged culture with high nutrients for rapid growth to achieve maximal cell density. And Stage 2. Once the stationary phase begins, key nutrients, carbon, phosphate, nitrogen, and specialized side chain precursors like phenylacetic acid for penicillin G are tightly controlled by slow, continuous feeding to maximize secondary metabolite production while preventing the growth repression we just discussed.
That's a lot to manage.
That is a fine art.
We've covered a remarkable amount of ground today, confirming that microbial secondary metabolites are so much more than just antibiotics.
We really have. We saw that fundamental difference between specialized, non-essential secondary metabolism and primary metabolism. We mapped out the crucial mechanisms of action from the irreversible, elegant suicide inhibition by beta-lactams on the peptidoglycan wall to the polycationic destruction and misreading of ribosomes by the aminoglycosides.
And the central narrative throughout this deep dive has been that constant, escalating evolutionary arms race. We saw how this war is fought using every tool available, from the rational chemical design that created debecicin and amikacin by building molecular shields,
to the genetic engineering of modular polyketide pathways that promises endless new chemical scaffolds. The complexity is just staggering, and the geographical data proves that resistance is a direct consequence of human usage.
Right. The emergence of constitutive mutants fighting off fourth-generation cephalosporins The rapid development of single-channel loss resistance to carbapenems, it just confirms that the microbial world learns faster than we can invent.
It adapts at an incredible speed.
Which leaves us with this final, perhaps provocative thought. Since the scientific challenge is no longer just finding new drugs, but managing the efficacy of the ones we already have, the fate of modern medicine rests so heavily on public health policy.
It really does.
The direct correlation between antibiotic usage patterns and resistance prevalence means that every prescription written, every feed additive used, and every hospital strategy for drug rotation or recycling is a decisive action in this global war. We can invent a new drug, but only cautious, responsible use can preserve its life.
The microbial world will always adapt. We just have to be smarter and faster in managing the tools they gave us.
Thank you for joining us on this deep dive into microbial biotechnology, and we hope you walk away better equipped to understand the crucial field of secondary metabolites. We'll see you next time.